So hi, hello and welcome. Microbehunter here and welcome to another Saturday microscopy live stream. Well, actually today it's not a live stream. It is this video here is pre-recorded. For private reasons, I'm not able to appear here live, in order to maintain the regular frequency of the Saturday live streams, I decided to pre-record it. But you are able to post questions and to communicate with each other because there is a chat function on the side. And if you also have questions for me, well, then I'm not able to, unfortunately, answer them live. But later on, I'm going to collect the questions and then I'm going to try to answer them in an upcoming episode. Well, as traditionally, welcome all around the world. I suppose that some of you have already started to post in the chat box from where you are and have been sending greetings all along. And what I would like to do today in this somewhat shorter episode today is I would like to, not only put this decomposing piece of wood under the microscope, but I decided, because many of you already seen similar things before, I decided to talk a little bit about my background in microbiology and what I've been doing many years ago. Okay, so basically, I'll be talking about that, and then at the same time, I'll be preparing a couple of specimens for microscopy as well, simply because I want to know what I can find here. Okay, as a short intro though, before I actually start doing these things, is maybe you notice this orange little blob over here. I've actually made another video and published another video a few days ago in this channel here where I put this orange blob under the microscope. I was kind of hoping to find a slime mold. Unfortunately, it's not a slime mold, but it's still an interesting fungus called Tremella. It's also known as the yellow brain fungus. Not the orange brain fungus, as I've mistakenly said in my previous videos. I'm also going to put this a little bit under the microscope. Maybe we're able to see a couple of spores again, right? But essentially there's no point for me telling you what I'm doing because you can see it in any case. Over here I also have this was the water where I put the wood in, there's also some stuff in here, so I'll also put that under the microscope, and we're going to see how far we're going to get and how long it's going to take us. Okay, so what I'll be doing right now is I'll simply tell you a little bit about my personal background. Many of you know that since 2018 I've been making YouTube videos on this very channel here, and microscopy YouTube videos, because I'm basically I've been interested in microscopy for a very long time, obviously. So I decided back over 10 years ago, I guess it was already, to start publishing a magazine. I called it the Microbehunter magazine, and I've been publishing that for a couple of years. I've been basically doing everything. I collected articles from the community. I did the layout, and then I also published them for freely available. If you want, you can still download them from the website. But this was very, very, very time consuming. And for this reason, I decided not to do that anymore, and to move over to YouTube and to do YouTube videos. So this was actually also very enjoyable. I like making videos and observing and so on. So for this reason, this was quite nice. And essentially, ever since then, I've been steadily growing my community. And people got more and more interested in microscopy, I hope, at least. Also because I've seen that many more people actually started to also upload videos on YouTube. Essentially, it was basically an important thing. Also, I think for Reddit, for example, is another thing that there are many posts. People started posting more, and this kind of also motivated me a little bit to dive in also deeper into microscopy. Okay, and what we have over here is this water from the wood, decomposing wood, and we can see a couple of living things here, right? Yeah. So this is basically a little bit the history of the channel. Almost in parallel, I think a year or 2 years later, I also opened my second YouTube channel called Microbehunter Microscopy, where I talk a little bit more about the technical details of microscopy, because a lot of people started asking me questions, and instead of simply answering them in the chat or in the in the comment section, I decided to sometimes simply make a separate Q&A video, and you can also watch those, right? So this is basically something that I've been doing, but actually this was not, microscopy was only a very minor part in my training. I basically, from my background, I've got a master's degree in research microbiology. I was mostly researching for my thesis bacteria, not mostly, actually exclusively. So protists, protozoans, and fungi, and so on, as you see, as we see here quite often, were actually not things that I studied, right? So it was actually, what I've been doing is I've done biochemical tests on different bacteria, genetic analysis of bacteria to identify what they are. Yeah, and essentially, microscopy is not really needed so much for that, because all of the data, or most of the data, is collected by doing analytical chemistry. So I've been growing bacteria, I've been isolating them, growing them in pure culture, and performed a variety of biochemical tests on them. And depending on what the bacteria were made of, you can kind of narrow down what they are. And this is quite different when you want to identify unknown bacteria, like I've done, compared to known medically relevant bacteria. Because if the bacteria are medically relevant, then you have a whole a limited set of protocols that you can apply, and then you can quickly identify them and narrow down what they are. And sometimes it's not even necessary, because all you're interested in is curing a certain infection. But essentially, if you are studying unknown microorganisms, because the biodiversity is so large, you have to very often optimize the methods, because sometimes they don't want to grow in the growth medium that you prepared. And there was always a lot of optimization, method optimization had to be done, right? And essentially, this is what I've been doing. I got assigned a whole bunch of bacteria, which we collected, and my job was kind of to figure out or narrow down what they could be. Yeah. And I said microscopy was not a very prominent part, because microscopy was used more as a quality control. So if you put some bacterial culture under the microscope, and if you saw different shapes of bacteria, then you know that it was probably contaminated. So it was all used for characterization, but you cannot use microscopes alone simply for identifying bacteria. You cannot just put bacteria under the microscope and then you know what it is. It's not going to happen. It's not going to work, because bacteria, which are very different, chemically, very different, and basically can appear to be very similar under the microscope, and vice versa. Yeah. So from looking at organisms, you cannot, especially bacteria, you cannot automatically conclude that they're related. Just because two cars have the same color, that's my analogy always, just because two cars have the same color does not mean that they're the same brand of car, right? To use an analogy here. Well, that's basically what I've been doing for approximately 2 years during my diploma thesis. So it was essentially what you called systematic bacteriology. It was called, which was a little bit unusual in the microbiology department where I studied, because most people who did microbiology essentially were involved in mostly molecular biology. So they were doing a lot of DNA studies, DNA analysis, genetic engineering especially. Also, they tried to isolate certain, maybe proteins that they expressed in bacteria and so on. So basically it was much more DNA based. And my thesis was a little bit different in the sense, let me switch to a higher magnification, was different in the sense that it was more analytical. Yeah, and that was also a little bit more in my own preference. So I'm right now using the 40 times, and you can see that in this piece of wood, water, you see a lot of different microorganisms. Yeah, so basically what I've done, in case you, because people ask me, where can I find those microbes, and what do I do, and where can I find them? And I don't have a pond next to me. So what I've done is I've just simply taken this piece of wood here, this piece of wood, was a dry piece of wood, okay? And I simply put it into a little bit of water here. Not completely immersed, and only a little bit of water on the bottom, right? And then the wood was kind of, soaking up the water. And some of the wood came off, and that's why the water looks the way it does. Yeah, it's very, very greenish, and So it was regular tap water, by the way, non-chlorinated. So if you don't have that, you can always, of course, use bottled water. That's also recommended. Yeah. And where is it? Here we go again, right? And then you're able to see. So some people, let me talk a little bit more about the things that I see here. Some people ask me about the color. How come I'm able to change the color here? That is because I've got a special equipment called DIC. Most of you are able to see it, yeah, like this, yeah, which is a regular bright field. Yeah, if you want to have colors, you can use color filters. Yeah, but that is would be bright field. Essentially, you're able to see everything in bright field that you're also able to see in DIC. The reason why I've got the colors is simply because I'm making YouTube videos and people also want to see a little bit more colorful things, right? Yeah. So yeah, I'll just put the maybe I have to switch this over here. Just a second. Let me. And the problem with this is that it really uses a lot of light. Okay, that's a little bit one of the disadvantages here. Yeah, another thing that I just want to say, you see those long things here? Yeah, this looks like a little bit more like I don't know, a hair maybe. Yeah. But sometimes, very often, you're able to see textile fibers and lots of microplastics, usually in fibers. So that's something that is almost ubiquitous these days. But this one doesn't. This one actually looks more like some kind of fur or hair, and not like a fiber, not like some microplastics. Yeah. Yeah. So this basically means that after when I did my thesis in microbiology, which I really learned a lot. This was quite nice. I liked it a lot because this was fairly biological in the sense. So I was essentially isolating bacteria. I was isolating bacteria from the air, so it was an air quality assessment, and I had to grow them in pure culture. That's the first thing that you have to do, is you have to somehow bring the microorganisms into pure culture so that you are able to analyze them individually, right? And this was essentially quite time consuming because not all bacteria like to grow in pure culture. Sometimes there are complex associations where some bacteria depend on other bacteria as well. Yeah, they have complex, basically communities, right? Essentially, once I've done that, I did my biochemical analysis. Then I had to dig through a lot of publications and papers to kind of narrow down a little bit into which direction it could go. Yeah. So this is basically it was the end of my studies. At the beginning of my studies, it was like this that. It was lots of general biology as well, so I had to do courses in botany, in zoology. They changed it in the meantime. Depends a little bit on the university. They changed that because molecular biology and genetics is such a vast and complex area that sometimes they start diving in directly into chemistry and cell biology. But I still had to take courses in the more general biology subjects as well, which I kind of like because it kind of broadens a little bit the view, right? So so this is basically what I've got here under the microscope using this water here. So what I'm going to do, however, is the following. I'm going to try something a little bit. I'm going to still use the same water, but I'm going to really go down with the amount of liquid here because I want to limit the movement. No, you know what I'm going to do? I have a yet better idea. Okay, let's see if this is still okay. Look what we got here. Beautiful. A couple of weeks ago, as of the making of this video, I don't know when you're watching this video, but just maybe you can go back into a couple of weeks ago in the live stream section. I made, this is methyl cellulose. I made some methyl cellulose, which is a very viscous substance, which limits the movement of protozoans. Yeah, because sometimes they're a little too fast. So what I'm going to do now is actually it's pretty cool. I didn't think of that. I almost forgot this tube here. So I'm going to again take a little bit of this liquid over here, and let's take a little bit of this liquid again, from this water, right? And let's put this away. I'm kind of worried that it might tip over. And another thing that I would like to do is I would like to take a little bit of this methyl cellulose and I'm going to mix it in with those here. The reason being that it's so viscous that it's going to limit the movement of the cells. And then, if this happens, they should be easier to observe, I hope. The only problem is it's really foaming a lot. And I don't need a lot of cells. I just want to have a few of them that I'm able to observe properly. Another way of limiting their movement is to kind of squash them between the cover glass and the microscope slide. So here, usually when you apply the cover glass, the bubbles are pressed apart anyway. So let's have a look here. It's again a little bit, always a little bit of a, how do you say, surprise what you're able to see. So, yeah, they're still moving around quite quickly and too bright. Okay, yeah, you see over here the liquid spreading apart. Okay, I need to let's go up a little higher. So it's also a question of kind of quantity. Maybe I didn't get a lot, or maybe I'm not really in the focus level. Here we go. Ah, yeah. Yeah, they're still moving quite quickly. So maybe I didn't take enough of the methyl cellulose. I don't know. Yeah. Again, a lot of fibers here. Again, it doesn't mean that these fibers were in the original sample. It could be that it's some kind of dust that was collected, on my microscope slide. Yeah, but this one over here seems to be moving a little bit slower. The good thing about the methyl cellulose is that it does not change the osmotic balance, and therefore the microorganisms, they do not dehydrate. So for example, if you use glycerin, for example, then this is actually will take away the water from the cells, and they're going to shrink and they're going to die. This methyl cellulose doesn't do that. Yeah, I don't know if they're moving that much slower. Yeah. It's also interesting when you take samples from a jar, different regions of the jar actually have different microorganisms depending on the oxygen concentration. You know, this guy is going in a circle. So let's simply try a little, over here. So let me see. It seems to be caught up a little bit with something here. Yeah, I mean, you get the idea, right? My suggestion is to simply keep experimenting and not use too much water, but if so that the vertical movement is limited. Okay. So right now it stays mostly in focus because it cannot move up and down so much, and this methyl cellulose also slows them down a little bit because of the high viscosity. Maybe I should have taken a little bit more. Yeah. So what I would like to do now, I just see that there's one issue that I have, and that is that there is some liquid over here. So this means that the slide sticks to the over here to the stage. So I'm going to quickly wipe it off. You can just see what I'm doing here on the top, because sometimes a little bit of water spills and then it's very sticky. Okay, so also over here. So let's try it again. Okay, here we go. And let's try to focus and find something interesting. So what I would like to show you now is let's have a look. Let's make it a little bit brighter here. In the background, you're able to see those tiny dots here, and I suppose that there are many bacteria in here as well. Bacteria can be seen quite nicely under phase contrast, so I've got only one phase contrast objective connected here. And let's see if we're able to see something here. I need to open the condenser. Everything's a little bit out of focus again. Yep, and indeed, you are now able to see those tiny things in the background. Yeah, many of them are most likely bacteria, especially if it looks like it's like a rod or a small chain. Yeah, and those bacteria are actually transparent, but phase contrast makes them appear darker on a brighter background. Yeah, you can also see that they seem to be sticking to the glass surface, and that the protozoans, the larger cells, they're kind of swimming on top of it away, and therefore they're a little bit blurry and out of focus. Yeah? So that's quite nice, I think. What I've not seen so far are rotifers. I've not seen any. I have also not seen any nematode worms yet. Okay, so it's not a, yeah. So this basically shows that in different samples we have different organisms here. Quickly also close this again. Yeah. So just wanted to share this with you in case you don't have a pond or anything next to it. Try searching for a dry piece of wood. Okay, put it into a jar for a couple of days. Make sure that it's not completely immersed, so there must be a lot of oxygen present as well. Yeah, I think they do move a little bit slower, especially this one over here. People want to know, okay, how can we identify them? There are identification books available, and very often those identification books, as I found out, is you find an organism, you find the organism, but often it looks only very similar, but not exactly identical, because again, biodiversity is so large that chances are pretty good that you're also going to have some species in there probably that are not in the identification book. So let's do the following, just for the fun of it. It's 21 minutes. Again, if you joined in a little bit late, this video over here has been pre-recorded. So this means that if you type in something in the chat, then I'm not able to directly answer it. Okay, but please feel free to do so anyway, because if there are questions in there, then I'm going to try to address them in an upcoming episode. Right. So let's do the following. I'm going to switch over again to the desk. Actually, I decided I wanted to talk a little bit more about my, about my university studies. So one of the things that I've been doing is I used a device called a gas chromatograph. And that's a really cool device, pretty expensive, and I liked really working with it, because it was interesting. It was like this, that my job was as I mentioned, to analyze the composition of the bacterial cells. What chemically are they made of? And among other things, I used to analyze the composition of the cell membrane, of the bacteria. In order to do that, I had to extract the cell membrane from the bacteria. So you kind of collect the bacteria from the jar that you basically from the medium that you were growing them in, and you basically did a whole bunch of extractions, and then you took those, basically you removed everything else from the bacteria except those the lipids of the cell membrane. And then I injected those into the gas chromatograph, which is a huge device, and basically which connected to computer. Yeah, and then based on the composition, on the type of lipids present, it was like this, that they were then separated by the gas chromatograph. The phospholipids, the fatty acids, basically, of the lipids of the membrane. For those of you who are a little bit into chemistry, essentially, I analyzed with the gas chromatograph, and then basically I got a diagram with peaks, and then going up and down, and then I had to kind of identify which peak is which fatty acid. Then based on the pattern that I got, I could then dig through a whole bunch of publications, and then figure out probably which bacteria it is. Yeah. So you see, it's a very different way of actually trying to identify bacteria compared to the use of, let's say, what many people assume. Why can you not identify them using microscopes? Yeah. And I'm saying is that unfortunately microscopes do not provide the level of detail necessary to identify bacteria. All the way down to let's say to sufficiently high degree. Microscopes simply are very important, very valuable, but they're not able to solve all of the problems. Like no method is able to solve all of the problems. Yeah, so I hope to be able to find again different organisms here. Yeah, here we have the challenge that there are a lot of wood particles here and so on. So what I'm going to try to do is I try to dissolve whatever is in here away. I need a little more water. And then I'm going to show you what I'm going to do in order to remove those parts here. because if I put a cover glass on top here, it's going to be way too thick. So we're going to try the following. Look, I'm going to try to move everything a little bit to the side. Okay, and I'm going to, yeah, so, and then maybe with a liquid over here, pop. We're going to do it like this, okay? So that's kind of my hope that, yeah, the big particles are here, and microorganisms probably are going to be in the liquid part here, because I want everything to be flat, yeah? So let's give it a try. Again, I have not tried this yet. So that's still the old slide here, and let's go for the new one here. Maybe we'll go down with the magnification again. And yeah, here we go. Let's see. Yeah, it's also quite something going on here. Now this here, that's now a rotifer. Okay, why rotifer here? Of course, because they like to attach to a surface, and on the wood, that's of course a surface, yeah, where they can attach to. So at university, this was quite an interesting thing. It is a totally new experience, obviously, if you compare it to school. Because at university, when you start university, that's the real thing. You know, it's not this, you're doing real experiments, right? And not these, under quotation marks, Mickey Mouse experiments that you do in school. I'm a teacher right now, so I think I can say that. The real stuff means you actually get to work with really expensive lab equipment that actually does need some training, because they are used also in research, and they simply also make them available for students. The chemicals that you use there are incredibly expensive. For example, I used to work with doing molecular biology with so-called restriction enzymes. These are basically enzymes that cut DNA, and they actually come in a tiny, I don't have one here, they come in a tiny tube when you buy them, in a tube like this, yeah? And when you order them, you pick up the phone and you say, I need this and that enzyme, and the company, and then they'll send a courier service. It's a guy on a motorbike with a huge styrofoam box filled up with dry ice. Because it has to be frozen all the time, so it's not delivered by regular post, but by individual delivery. One of these tubes costs a fortune. Unbelievable, you won't believe it. And then what you do is they deliver this in person. You have to pick it up. The guy comes in and you pick it up, and you have to immediately put it into ice again. It was like this, that we were told that one of the students there essentially looked at it, took out the tube, looked at it, says it's empty, and threw it away. And it wasn't empty. The liquid was so little that you couldn't see it, right? So they spent quite a bit of time digging through the trash can, trying to find it again. So these are the anecdotes, just to show you a little bit of how work in a molecular biology lab is different than for example, doing microscopy. Yeah, you're working with enzymes. That's why you need those specialized pipettes. Yeah, because these microliter pipettes, because the amounts that you're actually taking out are so small. Yeah, so this is one of those things that we were kind of warned to please, be careful. Sometimes the liquids is so small that you're not able to see, but it's still there, and it's costs a fortune. Yeah. In another case, there were these anecdotes that were told at the university, and that is that this is kind of the horror stories that they kind of warned us that at the beginning of a research project, a student got a E. coli to work with, which is a bacterium, right? And cultured the E. coli bacterium and to do the experiments with. Yeah? And the experiments didn't work out. For months, and at the end, they turned out, somebody put it under the microscope, and it turned out it was not E. coli at all. The student accidentally continued to work with a contaminant, some kind of a yeast or something, totally different. Totally different. So basically, months of work for nothing. Because the students, as this subculture did, they took it from a petri dish and they transferred it to a new one. They instead of transferring the bacteria, they were transferring accidentally one of the contaminants. right? So basically, you couldn't get any result, and this actually quite happens quite easily. And that's when we microbiologists said is, you should have actually put it under the microscope. Yeah, so microscopes are still good for something because they're important for quality checks. Why didn't you use a microscope to do a quick check? Then you could have saved yourself a couple of months of wasted time, so to say. Yeah. Yes, again, lots of microorganisms here from the wood as well. Okay, so yeah, but interesting. In the previous wood, I found a lot of nematode worms, but not so much here. Occasionally, what you're able to see also, they're difficult to find because they move so slowly, are amoeba. They change their shape, but sometimes they are a little bit difficult to see because they're not so evident because of their slow movement. Yeah. Yeah, so let's see. Let's continue to watch here what we're able to find here. Yeah. Yeah. So this. Yeah. One of the other interesting things is that concerning the biodiversity of bacteria, the majority of them can actually not even be cultured, in the lab because we cannot create the appropriate conditions, right? And so they've done. How do they know that? Is because they've actually done DNA studies by directly analyzing the DNA from a sample, and they found out that the DNA in a sample is way higher than the actual number of organisms that you're able to see there, right? Or that you're able to culture. And for this reason, we can conclude that it's sometimes in the lab extremely difficult to create those conditions that you're able to grow those microorganisms that you want to grow. Not a problem here in this case is because we're just observing them, but let's say if I wanted to subculture one specific organism, it might be quite difficult to do that if you don't know what the nutrition requirements are. Yeah. And over here, in a natural sample like this, with the decomposing wood, well, only those microorganisms will multiply that actually are able to deal with the current situation, with the current environment. So I don't know. Pretty much, I would say pretty much almost the same ones as we had before. Occasionally, occasionally I was able to see some amoeba as well. And normally, what I'd have to do is and that's I don't have the time right now. Is that I take a video of a certain scene here, and then I speed it up, and then you're also able to see the movement of the slow moving microorganisms. Yeah. But they are quite. Yeah, it's I'm using the 10 times objective, so let's go up a little bit more. Yeah. So this one here, as you probably have already seen, is I did not add any methyl cellulose. Yeah. So, I mean, if you have any specific questions, please do feel about let's say studying biology or microbiology. Please feel free to post them in the chat. Again, I'm not able to answer it live, but I'll collect the questions, and then in an upcoming episode, I'm going to respond to them because this video here is pre-recorded. Okay. And in many cases, I don't know what you're seeing here. Sometimes it's debris that you're seeing, fragments. Yeah, this one, for example, very clearly a rotifer, simply by the way it looks and the way that it moves and the way that it attaches to a surface. Yeah, so and very often what you also see is that over time there is a progression of different microorganisms appearing and disappearing. So also depending very much on the oxygen concentration of the sample. Some organisms are more oxygen tolerant than others. And generally, if you have a sample and you add some food to it to do an enrichment culture, you got to be a little careful because bacteria like to multiply very quickly, and then what they do is they will really push down the oxygen content of the specimen, and then also the other microorganisms start to die off. So don't overfeed. Yeah. But this one over here is not an enrichment culture. It's just basically, yeah, from the decomposing wood. So, what time is it? Okay, I've been now talking for approximately half an hour. Seems to be not something of particular interest here. So what I'm going to do next is I'm going to quickly see if I'm able to directly, yeah, because stereo microscopy is also a thing. I just wanted to, where is this? I need to find the one for the stereo microscope. Now you cannot see me anymore. Now I'm back. Okay, so that's now the stereo microscope. Okay, so let's just put this also under the stereo microscope. This is sometimes also quite interesting because these are of course lichens, and here we've got, here this is again our tremella, this is also known as the yellow brain fungus. Huh? So let's see a little bit. Let's go out here a little bit. Yeah, so these are basically the lichens, and they can be, I think, also best observed under the stereo microscope. Yeah. So it's often, yeah, a little bit overlooked. Stereo microscopes really are very nice as well. They just allow you to observe different things. And if you have children, I need to reach over to reach the focusing knob of the microscope. If you have children, especially young children, stereo microscopes might actually be a better thing as a present than compound microscopes, because stereo microscopes are easier to use and you do not need to prepare specimens. Yeah? But obviously you're not able to see the small microorganisms that we were able to see with a compound microscope. Even if you zoom in, okay, then have to refocus again. Yeah, you're not able to see those small, yeah, tiny single-celled organisms, obviously. Okay, so just wanted to yeah, also mention that a little bit. Let's go back a little bit. Where is this one over here? I need to go back to the microscope view here. So let's go up a little bit with the magnification. And every time when we go up with the magnification, the light intensity goes down. Here we go. Yeah. And I have to be careful. I have some other things standing around here. Ah, yeah. Let's have a look. Let's have a look. I don't know. This is where I have to guess a little bit as well. Huh. Did you see those long things here? What could they be? I mean, they look a little bit like they could be spores of a fungus as well. Fungal spores or conidia. I would say they're probably too big for bacteria. Yeah, but yeah, that could be one of yeah. Could be this could be fungi. Yeah. Let's do the following. Maybe we can. Yeah. I need to go down a little bit. I need to do this. You see, this imaging technique uses a lot of light. Yeah? Let's see if I'm able to. There's a little adjustment ring that I can also turn to compensate for the cover glass thickness. Here at the top. Yeah. I mean, you get the idea. I don't know. Sometimes, do you see this one over here? This could be an amoeba, but I'm not quite sure. Yeah, it's not moving at all. But sometimes they have this irregular shape, and the circle that you see in here could be the nucleus. One would have to watch now if it's actually moving. But I think you get the idea why amoeba are sometimes very difficult to see. It's because they move so slowly, right? I mean, it does change its shape a little bit. Yeah, see, it does change its shape. Look, it's kind of going down now. It's also not. It's actually not one of the bigger ones. Yeah, it's changing its shape, so that's most certainly one. I wonder if it's only the lower part here, or if it's the whole thing. Yeah. So that is, yeah, it's moving. You see, that actually shows a little bit why it's a good idea to sometimes simply make a video and then kind of play it faster. Yeah. So that's definitely an amoeboid cell, and yeah. What else do we have here? Yeah, I'll go on for a couple of minutes, and then I'm going to call it quits. After I've finished my master's thesis, I decided to change jobs. I decided not to stay in education, and I decided to become a teacher. Not to stay. I have to correct myself. I decided not to stay in research, and decided to go into education to become a teacher. And I had to restudy. I had to get a teaching qualification. I also studied then computer science as a teaching degree. And I very quickly discovered that many of the things, the interesting things in biology, it's very difficult to do in school because you've got a curriculum to follow, you've got a whiteboard or a chalkboard, and then you have to explain cell division, right? The steps, yeah. But observing the things under a microscope, even though we have equipment in school, many schools have microscopes, is sometimes difficult because it takes practice. And simply showing slides, you might as well just, I don't know, yeah, you don't see movement, right? So very quickly I realized that I would like to share more about biology with my students and to the general community. And sometimes educational institutions like universities and schools, they simply don't allow you to do that because of the infrastructure issue. And then you've got a syllabus to follow, and then you need microscopes, and is the lab free or not? And then by the time you've got everything set up, the lesson is already over, right? So that's what was also one of the reasons why I decided to, well, okay, if I cannot do it in school, then why not? Not so easily in school, then why not make videos, right? So this one over here also seems to be an amoeba. Yeah, we can also watch it a little bit of whether it's able to change shape. And so what I'm doing right now, ever since I've been making microscopy videos, I was basically rediscovering a little bit the connection to nature. And I'm of course using those videos as well for education, because I of course show them also. And I know that other some other schools actually also show these videos. Yeah? This one over here is quite, I'm almost quite certain this one, these are the spores of a fungus here. Yeah, conidia, so called. Yeah, but those long ones also kind of look like that. Yeah, so yeah. So let me see. Yeah, not so much movement here. So the question you might wonder is where are all of them? Where are all of those moving cells that we just saw a couple of minutes ago? And sometimes what they do is they follow an oxygen gradient, and sometimes they can be found further towards the edge of the cover glass. I don't know if also in this case over here, but usually they collect somewhere in a place where the conditions are a little bit more ideal. Let's go down with the magnification again. Yeah, and yeah, here, for example, here on the side, I'm now reaching the edge of the cover glass here, right? And that's where you can find more of them, because they want to go there where there is more oxygen. So if there's some algae inside the sample, some green algae, then this is less of an issue because they produce oxygen. Yeah, but usually after a couple of minutes, if they're all gone, check the sides of the of the microscope slide. See, that's the edge of the cover glass, and here we're already outside. Yeah, and here this is where you can find all of them. And if we move further to the center, look here, that's not the center. Much fewer. Yeah, because they avoid the low oxygen area and they move towards the side here, towards the oxygen, and here they are. Yeah. It's a very common phenomenon to observe. It kind of shows also how living things are able to respond to environmental changes. Yeah. Well, I nearly forgot. I did promise you to put this fungus here under the microscope as well. It doesn't quite look quite as nice anymore as a few days ago. Yeah, the green things here, these are of course the lichens, as already mentioned here, but that's the fungus. Yeah, and let's simply make a quick slide here, and let's put it also under the microscope. Yeah, because also I'm interested a little bit in how the things look, how the spores look like. Okay, so let's do this very quickly, and it works like this. As always, I recommend that you put a little bit of water, I'm always exchanging the tips here. I know it might be a little bit of a waste of plastic. I've got here a whole bunch of replacement tips. And over here, this is my trash can. Yeah, this is how the trash can looks like. Yeah, I know, but one always has to be environmentally aware. I am aware of that as well. But still, I do not want to do any cross contaminations here. So now what? A little bit of water here, just regular tap water, and let's simply take a few of. It looks feels. You know what? I'm going to go down a little bit so that you see this better. The camera is shaking. Here we go. So, and let's see if I'm able to grab a little bit of this here and suspend it. Yeah. I forgot to rinse the tips of the tweezers. It doesn't matter. Yeah, because I think. Yeah. And let's have a look how this looks like. Sometimes it helps to lift it up a little bit and see. Yeah. I don't know. Yeah, it does turn a little bit cloudy, which is a sign that there should be spores. Something should have come off. This one over here, this piece of the fungus is. Oh, I tipped over my cover glasses as well. So let's do the following look. Just going to use the cover glass to kind of take it down here. Here it is, huh? So, yeah, let's have a look. Okay, yeah, there is something in here. I mean, you might not be able to see it directly here under the camera. Okay, but yeah. Let's have a look. Okay, so I need to switch over again to the microscope, and here we go. Yeah, too many of them, right? So that's still part of the fungus of this yellow blob. And what do you see? All of those things here, these should be probably the spores. Too high of a concentration. So let's go further away here. And the magnification is also fairly low. So let's go up with that as well. Okay, let's go back here. And yeah, here are different. Here, fewer. I have to find a place where it's actually better to see. I think I might be on the wrong focus level, or not. Lost the focus. Here we go. That's it. I see I lost the focus. So 40 times. And let's open up a little bit. Yeah, here we go. Okay, so I don't know, countless spores, and of course. That's the reason why some people become a little bit allergic against certain fungi. Not all of them, but certain fungi, because of course they spread over the air. And what they do is they might cause certain reactions. And here, this is actually the thing that made me realize that I'm dealing with a fungus here. Is because those tiny lines that you see, excuse me, yeah, these are the cells of a fungus, because they're long and thin, and yeah, the so-called the hyphae. I've been informed that I've mispronounced this in the last in a couple of videos. These hyphae are, of course, the cells of a fungus, and you know what? I think we just for this here we just switch again into phase contrast. Yeah, and sometimes. Yeah, phase contrast allows you to see them also a little bit better. No, actually, in this case, not really very much. Okay, I think the density of the of everything is a little too high here. Yeah. And here we go again. Okay, let's turn the arrow off again. Yeah, so there are plenty of things to discover. Yeah. Plenty of things to discover. And normally what I do is I also need to say that because sometimes people ask me, how come you've got such to contrast the images. And I tell you, in most cases, not in this case right now because I'm directly recording it, but in most cases, I actually use a video editing program to improve the contrast a little bit. Yeah? So these are again these are the cells of the fungus here that you see. Okay, those long thin ones over here. Yeah. Now they're all moving because by capillary action spreading beneath the cover glass. Yeah. I just wanted to share this with you as well. Let's go here again 60 times. Yeah, I need to switch over to the correct. Yeah, and here we go again. That's too much. Yeah. And here we go again. Yeah. So many spores. You know, folks, what I'm going to do now, I think I'm just going to call it a day for today. I think, as again, if you've got any specific questions that you have, please feel free to post them because this is a pre-recorded video. For those of you who just joined in, I'm therefore not able to do a live Q&A session. But I hope that still you had possibilities to exchange some of the views and experiences with the community by using the chat function on the side. I'm going to now, basically all I'm going to do now is I'm going to leave it for today. I wish you all the best. I hope that you still and that you enjoyed this video over here. Got plenty of other videos as well in this channel. So if you go back into the previous into the history a little bit, you can see that there are quite a few also live streams available. So I hope there's something in it for everyone. I'm going to say goodbye for today. Happy microbe hunting. See you around next time. Okay, bye-bye.